Today with me, I have a special guest. His name is Philipp Dexheimer. He's a scientist and a science illustrator, science artist, which is a career that I have not yet begun to explore. And I hope that he can tell us more about that career path as well as his scientific journey. So I'm very happy to have you on, Philipp. How are you doing today?
Hey, thanks, Kenan, for having me. My pleasure to find the time to have a chat today. I'm good. It's Sunday in Vienna. Autumn is coming around the corner, so time to drink tea and get cozy. And looking forward to have a nice chat. Yes.
Awesome. Your energy is awesome. You just lit up my Sunday here. So that's thank you for that. So you did your PhD and postdoc at the Institute for Molecular Pathology in Vienna. And from what I understood from the couple of minutes we chatted before, you just recently, a couple of months ago, full-time switched to being a freelance scientific illustrator. So maybe tell me first, how did you get into science? How did you get to do what you were doing during your PhD and postdoc? And when did you start entering this and exploring this field of science illustration?
That's a good question. So basically, regarding the science, I figured I always found fascinating, especially biology or life. And when it was time to decide what to study after school, a couple of options were on the table. I thought about architecture for a while, so I always liked design as well. But it ended up being biology because I figured it's really trying to understand how life works was one of the things that fascinated me the most. And biology seemed like the right field to follow up on that interest. And then in the beginning, I didn't have a very concrete idea where to go scientifically. And after a couple of semesters, RNA biology struck me as really fascinating. So it started with, the multifaceted roles of RNA it plays both in our cells and in the history of life on planet Earth. So I was attracted by RNA biology. And then I went to Vienna for a summer internship in the lab with Luisa Cochella. And for the first time, got to work with C. elegans. And this was really quite something. I love organismal biology. So, cell culture is very interesting. It has its own domains. For me, organismal biology, it's a very, very exciting way to tackle questions because you end up working with the whole animal. And no matter what you study, chances are that you will come across many other exciting aspects of biology. And also seeing things in the context of the whole organism, that was something that I really wanted to do. So I decided to do my PhD using C. elegans as a model organism to study microRNAs in animal development. And the PhD project was about, when you think about how this very single cell, the fertilized oocyte, manages to divide and form all these different cell types and tissues and organs, and in the end make up a whole complex organism. That's, to this day, for me, mind-blowingly crazy. And we were basically asking, how do microRNAs regulate this very complex process? This was my PhD work. And in the end, I was not sure how to continue. I think many people who did their PhD can relate to this, or who are doing their PhD. It costs you. It's an exciting time. I love it. I also would do it again, but it's a lot of effort. So at the end of my PhD, I had to take a step back and take a breath in order to be able to really think clearly in an inspired way where my future journey should be headed. And for me, it was both. I love science, I love the lab, so both the academic career was appealing. I realized that I do have other interests, especially related to art. And during my PhD, I had this constant feeling that I wouldn't have enough energy and time to bring up for my other interests. So it turned out to be a somewhat constant struggle of you're really busy in the lab, there are these other things you want to do, and I didn't quite manage to strike the right balance during my PhD. So after my PhD, I decided to start a postdoc in the same institute. I also stick with C. elegans. We transitioned from doing RNA biology to more broad genetics, I want to say. And I figured during my postdoc, I want to set aside more definite time to work on my side project. So this is where I really started to work part-time as a freelance illustrator. So this was 4 years ago, roughly. And I would say I spent maybe 20-25% of my time doing illustrations or creative projects at rest in the lab. And this was at first for me the tryout period of when I do this professionally, scientific illustrations and art. Is this something that feels like a road I want to go down? And after 1 or 2 years, I figured, hey, I do enjoy this also professionally. That's always an interesting question. When you do something for fun, the moment you start doing it for making a living, your perspective changes a bit. And then 2.5 years into my postdoc, where it would have been the decision to really now focus full energy on the academic aspect or give it a shot with becoming a freelancer and doing art related to science. I said, okay, let's give this a shot. And here we are. So this was 1 year ago, pretty much exactly. I left the lab completely and focused full time on my own business and developing my skills. And it has been an exciting year. Lots of new changes and some unforeseen developments. And it's interesting to get into a new field and face problems which are very different from the problems you were facing in academia. So it hasn't been chill, but it has been quite a ride. And so far I can say I'm happy about the transition. Yeah.
We'll be right back to the podcast, but first, I want to say thank you to our sponsor, Litmaps. If you are struggling with the academic literature and you are overwhelmed by the number of papers you are trying to read and make sense out of it, Litmaps is here to help. And visualize for literature, you can find research gaps, and you can create a beautiful map and keep yourself updated every time a new paper comes out that is connected to the map you've built. And if you're working on a review paper with other collaborators, this map that you build, you can also share with your collaborators and have them build it so that you can go back and forth in exploring the important papers of your field. Litmaps is a fantastic tool that I've been using for over 6 months now, and I've found it incredibly useful for many purposes, and it's completely free to use. If, however, you want to use the more powerful Pro version, you can use the code in the description, Kenan30, and you'll get 30% off on all of your first-time purchases with Litmaps. Thank you, and let's get back to the episode. Awesome. So I think it's a very interesting career path and a very interesting journey that I think many people struggle with. I've met many scientists who are very creative. They have creative side projects. Some of them paint, some of them sing, some of them play an instrument or do something like that. But it's a very, many of them have this same issue of striking the right balance and then finding themselves at the crossroads, especially if they're doing biology. I'm not sure about other sciences, but biology is quite, especially wet lab biology, is quite lab heavy. By the time you're done with your day, it's not easy to be creative at the end of a day of under the microscope with your neck hurting and all of these things. So we'll get to all the new challenges you're facing with becoming a full-time scientific illustrator. But first I want to talk about your science a little bit and your PhD. You said you were working with C. elegans. For the non-biologists out there, this beautiful worm right behind you, which has many advantages to do science and study it. You studied it from the point of view of development, right? And microRNAs. Tell me, why did you choose that topic? And maybe what are some advantages of using exactly C. elegans to study these kinds of problems?
Thank you. It has been a while that I talked like pure science. Thank you for that question. So let's start with the advantages of C. elegans, maybe. It's interesting because it's a nematode, right? And especially to your non-biologist friends, it always sounds like a pretty obscure thing to do. So, you're a geneticist, it's interesting. What do you study? And then when you want to be a little bit cheeky, you hit them with we study worms. And that doesn't sound like the most thrilling choice of model organism at first to non-biologists. And it's interesting because nematodes are somewhat the opposite of humans in their biological logic, so to say. It's an animal which is as simple as it gets, if you want. So it's these tiny, tiny worms. They're 1 millimeter long, and they consist of around 1,000 cells. Every adult C. elegans consists of around 1,000 somatic cells. The nervous system has only 300 neurons, but it still qualifies as an animal brain. So for many questions, especially in the areas that we don't understand very well, it can be extremely helpful to start asking these questions in a very simple system. And the way biology works is the chances are you find out how the simple system works, you learn a lot of very important mechanics that you can also apply to more complex organisms like vertebrates. And from an experimental point of view, C. elegans is just brilliant. It's easy to manipulate. So when you have, especially when you do genetics, you always have to wait for the next generation until the genetic manipulations you introduce come into effect. C. elegans has a life cycle of 4 days, basically. So it's an amazing molecular playground to test your ideas. And it's fast. I always, when you think about mouse genetics, people have to wait for months. You have an idea, and then you have to design the reagents, how to test this. You make the mutant, and then it will take you a couple of months until you get the first slightest idea if, your experiment is going to yield something. And C. elegans, this is a matter of weeks. And you really work with the entire organism, and you can do beautiful microscopy in the entire organism. If you look at a plate of C. elegans, they're hermaphrodites, which means they can fertilize themselves. So you have a plate of isogenic individuals, which means they're all genetically identical, which from an experimental point of view is also amazing. You can really test two populations of which they come from the same mother, they're genetically identical, and it reduces any variance which comes from mixed genetic backgrounds, sort of thing. So I did fall in love a little bit with using C. elegans as a model organism. And it's also taught me more respect for, the little things in nature. After all, we have a very anthropocentric perspective, and people don't have a lot of respect for worms, generally speaking. And if you think about, I think it's 4 out of 5 animal individuals on this planet are nematodes, because they're just everywhere. Wherever on this planet where you can find life, you scoop up a pile of dirt, you're going to find thousands, if not millions, nematodes in there. So from their overall biological significance, it's also quite something. And lastly, the worm community, it's really beautiful. It's a relatively young field. So Sidney Brenner, famous molecular biologist from last century, he wanted to understand the animal nervous system. And he said, we are so far from understanding what's going on in the human brain that we should try to, understand the simplest animal nervous system there is. And for this, he was looking for the perfect model organism, and he chose C. elegans the worm. And this was in the 70s. And basically all C. elegans lab come from a lineage. They were trained by Sidney Brenner. I think I'm his academic great-grandson. And the field is very close. It's by now 10, 20,000 people, I guess. And people are very respectful. They don't, jump on each other's research ideas. So I always found the entire field is also a lovely community. Yeah.
I'm curious about some tools that one might use with C. elegans when it comes to, for example, genetics or studying these basic processes. So as a fly person myself, so I do understand that most people, even in the scientific biological community, don't appreciate very often these small organisms and the basic biology that can come out of it. But we who do it, we really do understand all of the advantages. So similar things to what you mentioned with C. elegans, we have, as you know, you work with flies as well. So you have these beautiful genetic tools. You have an organism that can still do complex behaviors, but not have a brain with billions of neurons, right? So it's way more simple. And there's all these tools that exist to easily experimentally manipulate the organism. So with the flies, it would be something like balancer chromosomes, which allow you to track exactly, just to look at a fly and know which chromosome did she get. which manipulation did this fly get, and it's easily to sort them through and kind of work with it. And then there's these huge stock centers that allow you to order just already existing manipulations. What I'm kind of ignorant of the tools in C. elegans. So what are some good things one can do in the worm that might be an advantage? I know that there's a defined number of cells and the defined number of neurons, more or less, so that can be an advantage. But what are some other things that one might utilize the little worm for?
You can freeze them, and this is amazing. So the Drosophila Stock Center you mentioned, they were basically working around the corner of my institute. So they had to flip 60,000 fly strains, I think every 6 weeks or so, right? So you had 4 or 5 people just concerned with flipping those flies. The worms you can freeze, and this is amazing because you don't have to maintain every mutant you make in the lab, but you just put them in the -80 freezer. You can thaw it up to 20 years later, probably even 100,000 years. There are some examples of worms being frozen tens of thousands of years after being stuck in the eternal ice, and they still come alive. So you can freeze them. Then what is the elegance specific? I think the flies and the worms, they have a lot of parallels, and in the end, probably Drosophila is the organism that taught us most about molecular biology from the animal realm, I would say. C. elegans does have the advantage of being hermaphrodite, so it's easier to, get homozygotes, you save one generation. Especially when it comes to the nervous system, they're not sure how the fly people go about it. In the worm, it's transparent, so you can do whole mount microscopy, and without any further manipulation, you can really scan the brain in 3D. And what some people do, they do brain scans and freely moving C. elegans, which I think, I'm not sure if fly people found out a trick here, but this would be one unique advantage, the small scale and transparency. I'm not sure if there is some definite techniques that set worms apart from flies. I would say genetic manipulation in terms of, you insert something into the genome by CRISPR, you inject a gonad directly. Probably it's a little bit easier to get a higher number of mutants. I'm not sure. I think they both have individual nuances where it's, advantageous to work with one over the other. I feel in general there are a lot of parallels. So I have a hard time to pick clear advantages from one over the other. One comes to my mind. For how could I forget about this? For developmental biologists especially, C. elegans has a stereotypic lineage, which means when you have a C. elegans zygote, you can basically, the zygote will divide in a stereotypic way and will give rise to these hundreds of embryonic cells, and every cell originates the same way in every embryo, so every cell has a name. So if you want to study development, in particular early development, you have this crazy unmatched advantage. You can really draw out the lineage from the zygote. Every cell has a name, and you can trace the ancestor of every cell that will eventually become a neuron or a muscle cell. You know the exact lineage and ancestors these cells go through. And this is, from a developmental point of view, extremely exciting because you can basically study in this embryonic precursor cell, which is going to turn into a neuron, what's happening in this cell way before it's a neuron that already sets it up for being a neuron later on. So I would say this is, from a developmental point of view, the one thing the elegans is really amazing for. The identity of every cell at every given time point of the animal's life cycle, basically. Yeah.
That's amazing. To just address the part about recording the nervous system, that's actually what I'm doing with the fly. And yes, indeed, it is tricky. You have to take out the brains, which is very difficult because of the head in, the cuticle that is quite hard, and the brain is quite sensitive, so it's easy to damage it.
It's difficult due to the fact that the brain is inside of the head.
Yes, that is the disadvantage. That is true. So it is quite tricky. So I could definitely see that as an advantage. I find this quite amazing. I know that people have been to freeze Drosophila. They were trying to develop these protocols to keep it alive. I don't know, does it work?
No.
But that's just quite mind-blowing that you can have worms frozen for however long and they'll just come to life with very long time after. Okay, so you work mostly on, by the way, you have a recent paper in current biology, so congratulations on that, and it focused on microRNAs and development exactly. Maybe can you briefly tell me why are microRNAs important in development? And what did you learn in this current biology paper?
So basically, microRNAs are a very interesting class of genetic regulators. For people who don't know, microRNAs are small RNAs, and what they typically do is they will bind the target mRNA, which is complementary in sequence, and then they recruit this RNA-induced silencing complex to the target mRNA, and they will reduce expression of the target mRNA. So basically, they're very small regulators of gene expression. And what makes microRNAs particularly interesting is when we ask ourselves what enables organismal complexity. So what makes a human more complex than a fish or a fly or a worm? Then people first thought it was genome size, where we sequenced genomes or knew how large they were. We learned that onions have about 3 times as much DNA as humans, so genome size can't be it. Then I thought it was gene number. When people learned by sequencing that, for example, flies have around 20,000 genes, flies have a bit 14,000, I think. C. elegans has 20,000 genes. The human genome has around 20 to 25,000 genes. So the number of genes can also not explain why some animals are more complex than others. Turns out the numbers of microRNAs in a genome is a much better predictor of complexity. Because in the end, what you need for complexity is you need many different cell types. So the number of cell types, it's a good proxy for complexity of an organism. And to make many different cell types, you need many different intricate regulatory mechanisms. So regulation of gene expression is key to complexity, if you want. So this is why I found small RNA biology in general very interesting. And now if you think about development in particular, what's curious, the elegans has around 150 microRNAs. And we know that most of them you can knock out individually without seeing a clear phenotype. So there are only a few microRNAs in C. elegans where if you remove them, you really get a striking effect. And this can either be due to redundancy between microRNAs, so you knock one out, but you don't see an effect because another microRNA, even if it's unrelated in sequence, can still, take over and repress the same targets. Or it could be that microRNAs have small effects, that repression of a target mRNA by a microRNA basically fine-tunes gene expression, but it's not a binary switch of this happens or this happens not. So it could be that while individual microRNAs have only small effects, removing one doesn't really mess up the whole system. And this was the question we tackled during my PhD. We have these 150 C. elegans microRNAs, and we know that only 2 families, so 2 groups of microRNAs related in sequence, those are critical for embryonic development. All the others you can take out individually, and the embryos are going to do fine. So we wanted to test the idea, is it that really these 2 microRNAs are all you need to make an embryo from a zygote? Or is this the only 2 ones we know, but there are many others which also play a role? And to test this, we had a funny idea, which in the beginning sounded a little bit too crazy to work, but we gave it a shot, and turned out we got lucky, or we got a solid educated guess. Our idea was we kill the microRNA processing pathway. So basically the enzymes which are responsible for producing every canonical microRNA, it's first a microprocessor complex consisting of drosha, the endonuclease, and pasha, the cofactor. Later we have dicer, and then gets incorporated into argonaute. And our strategy was to kill microRNA processing by removing pasha and drosha, so no canonical microRNA is produced anymore. And then the embryos die. So if you kill microRNA processing, basically your zygote will turn into a clump roughly resembling an organism, but it already fails to, fall into the worm shape, if you want so. And what we did is we killed canonical microRNA processing, and then we tried to reintroduce individual microRNAs piece by piece. And for this, we had to engineer an alternative microRNA delivery strategy. And like the small RNA aficionados will know there's a rare class of microRNAs called miltrons. It's a microRNA derived from an intron. So basically, you can, in particular in the worm, change an intron in a way that when it's excised by the spliceosome, it looks pretty much like the product of Drosha-Pasha cleavage. So you can produce a microRNA without relying on the canonical biogenesis machinery if you use the intron bypassing way. And with this strategy, we found out that if we put 2 microRNAs back into C. elegans via the intron bypassing strategy, where we kill the canonical microRNA biogenesis machinery, so these embryos are devoid of all microRNAs except the 2 that we put back. This way we could show that microRNAs, these embryos with only 2 microRNAs, they still make it to hatching. So all the major organs are formed, they basically form a larva, then they rest in development. So afterwards you need additional microRNAs, but 2 are sufficient for basically embryonic patterning and embryogenesis, if you want, so establishing all different tissues and organs. And this was the main message, but we don't mean to say that all other microRNAs are not important. Instead, what we think, what our results go in line with, is that you basically have a lot of microRNAs and only a few of them are involved in core developmental processes. So they will be really embryonic lethal if you take them out, while the majority of microRNAs acts more at the periphery of development. For example, if you have a given neuron and then you make a variant of these neurons. So this is often a process where microRNA is involved. Not having this won't kill you, but it will be critical for your survival in the wild. So this was the take home from my PhD work. Few microRNAs appear to be absolutely essential in animal development, while the majority of microRNAs appear to be active more at the periphery of development, regulating more nuanced processes instead of, very critical ones early on. Yeah,
That's quite beautiful. That 2 little molecules are all it takes to push an organism to some stage that master regulate many of these patterning processes. So if you take a parallel with higher organisms, and you mentioned before that there's this predicting power of the complexity of microRNAs and I guess their number. When you say a predictor for the complexity of the organism, and as I found out now, it's not genes, it could be microRNAs, would you expect that the number of crucial microRNAs in an organism is a better predictor than the total number for the complexity? Or how do you think about, in general, how microRNAs actually predict complexity?
that's an excellent question. I don't know the answer. I'm not sure if anybody does, because these types of experiments are so what we did in the worm, I think in a mouse, it would be extremely difficult to replicate. So in the mouse, we know of more microRNA examples where if you knock them out, you get more severe phenotypes. So it could be that C. elegans, being a fairly simplistic organism, is also more robust in developmental terms, and that the more complex you get, the more sensible different steps of development are to being perturbed. So I don't know the answer, and I'm not sure. I think it's something the field debates about since a long time. The lab of Dave Bartel or the Bartel lab might have something to add here. I could imagine that in more complex organisms, you have more microRNAs that play a critical role because the entire developmental process of a more complex organism is more sensible. So in a mouse embryo, which develops, not in a matter of 12 hours, but in a matter of a couple of days, and I don't know, I think it's 2 weeks, if something goes wrong, the downstream errors will be more severe. So this would be my take on, in higher organisms, you have more microRNAs which have critical functions because the entire developmental process is more sensitive to disruptions.
Yeah. I'm thinking in the moment, I would maybe expect it that they have like a different number of critical microRNAs and the number of ones that are modulating the peripheral processes that are not critical but are important for a neuron to do its proper function, for example. So they might grow with a different function. So maybe the number of critical one for every step of complexity, they grow, but maybe linearly. But the ones that modulate everything else grow with a faster function. So you would maybe expect much more. However, that's just it's a small model in my head that popped out. It doesn't have to be true at all. Your paper, and I think this gives us a wonderful transition point into your scientific art and scientific illustrations that you do. Your paper has a very beautiful abstract and a beautiful illustrated abstract. How do you go about creating a visual representation of a paper?
That's a very good question. It depends on the goal you want to achieve. Let's maybe stick with the graphical abstract example. So a good graphical abstract has different functions. One is that you really should be able to extract the essence of the paper, even if you are not from that field precisely. So I would say for this, it's important to isolate the singular key message that you have in your paper and try to visually represent this. Often it's a little bit complicated because especially more descriptive papers, they don't have a singular key message, but it's a series of results. But I would say number one is the goal of a good graphical abstract is that somebody who is vaguely familiar with this type of research understands what it's about. Goal number two is to increase the memorability of your work. I don't know how it's for you. For me, I often read a paper and then 2 or 3 years later, I think back about it because it's somewhat relevant for my research. And what my brain remembers often is a good graphical abstract. So if your memory has an image to basically tie the main message to, then it will be much easier to years later on, remember what was really the key message in this paper. So in terms of creating one, I would first try to isolate the singular narrative. What is the one key point your paper makes? If we found out that one, then either you have an intuitive idea of how to depict it. Some papers are very streamlined where, intuitively it becomes clear to you, okay, we have these two cell types we study and we do the same manipulation and it's two different outcomes, then it's an easy linear abstract. If it's a bit more complicated, then I would usually follow the systematic approach, which is a similar framework than if you write a paper. You have one panel or section which serves a little bit as an introduction, so as an overview. Let's say you study endothelial cells. In the first panel, you study endothelial cells in vasculature in muscle. So the first panel should be an introduction, showing people where are we located, what is the context we need to think about this. So you could show a muscle fiber, which gets smaller and smaller into the vascular or the vessels, and then you show an endothelial cell there. So first you establish the setup. What is the biological context we are thinking about here? Then I would establish the methodology. So what was the experiment done, or what methods did we use to study the question? I would have a third section, which is kind of the results, so the main results. And then a fourth, which I guess corresponds to the discussion to somehow what do we learn about biology here? So if I have to systematically come up with a concept for a graphical abstract, I would try to incorporate these 4 elements. What is the context / introduction? What is the methodology? What is the question and the methodology we used? What are the results? And what does this mean in the broader or big picture and biological context? That being said, it's difficult because on a case-by-case basis, you have different routes to get to the goal of making a solid graphical abstract. Yeah.
You mentioned already one advantage of having a good abstract, which is the reader has an easier time remembering it.
Yeah.
I have noticed in myself that when I read papers, and I think this is just a human bias, when I see a paper that, for example, it's a review, but it has figures and images that represent maybe some pathway, and they have colors flying around, not fitting at all and some weird shapes, and they're just ugly. I have a hard time, and I have to fight against this to still see the paper good, but I have a hard time convincing myself that the serious effort was involved in this if the figures are really ugly and not taken care of properly so that they are nice for the reader. Do you think this is actually a big deal when you scale it to thousands of readers, and who's going to cite your work and who's going to take your work seriously besides just remember it? How important do you think is the visual aspect of any scientific paper?
I might be biased, obviously. I think it's very important for two main reasons. So one you already mentioned is what do you infer from beautiful or bad-looking figures about the general quality of the work? And here we can be a little bit careful because also, as you said, it may happen, and I know a number of examples myself where you have a beautiful paper and it's amazing science, clean, well-done experiments, proper interpretations, but the authors just don't have a very strong visual sense for how to make it nice. So there is the case where you have a beautiful paper, but just doesn't look very nice. I think as humans, we tend to infer from every piece of information we get. We tend to infer how much effort did people put into creating this. When you write a motivation letter for a job, right, then one part is the real content of the motivation letter. The other part is how important was it to you that this looks good? How much effort did you put into the overall product? How much does it mean to you? And this goes along the lines of what you said. When somebody really seems to not care about the figures at all, then you automatically deduce that the entire product was not insanely important to them, because otherwise they probably would have put in the extra effort of, making it look nice. So I think on the one side, it's a general, you create an impression that whatever you touch, you work with a high standard on it, and you try hard to make this as good as you can possibly do. So there's that. The other thing is visuals intimately resonate with the human mind. This is in particular true for artwork, for illustrations, or scientific figures. It's a little bit less pronounced, but a good graphical abstract or a good figure, talking of color scheme and layout, it resonates with your mind. It basically nicely guides your mind through the information it's supposed to get out of there. And good illustrators, or good design in general, good design makes it easy for the human mind to follow along. It makes it causes you to not have to invest a lot of mental calories to figure out what do I see here. And brains love it. Brains love if you take them by the hand and gently guide them across a story. And in the end, what stories are, both orally transmitted or visual stories, it's vehicles of information. So if you think about a visual story in a graphical abstract is very much a visual story, then I would say it's a vehicle of information. And the better it resonates with the way the human mind is used to perceiving information, the more likely you will understand, and the better it will stick, and the more likely you will, remember this particular piece of information, paper, a couple of years in the future.
Let's say I'm a PhD student who is just working on a paper right now and wrapping it up, writing it, and I'm trying to make beautiful figures, beautiful abstract, illustrated abstract. And I don't necessarily have a budget for an illustrator, but I want to do a good job. Give me some tools to choose the right colors, to design properly, nicely, some principles that will guide me through that process.
So there are two parts of the process. One is the content part, and one is the visual part. For the content, you don't need any visual talent at all. You basically you can even do it with pencil and paper. You just need to decide what needs to go on there, what elements we need to have on the graphical abstract for people to be able to understand it. In this phase, I would recommend to get a lot of feedback. So, beta testing if you want. So, come up with a concept, find some of your peers, not your lab, because they know what you're talking about anyway. Find someone from your institute, close enough to your research that they would care, but far enough that a good test audience, and try to explain them your content draft of the graphical abstract. And then most likely you will have to revise it 2, 3, 4, 5 times until you, just from the content that is in there, have a solid basis that people understand what you mean. This is part one. And this everyone can do. You don't need to be a visual expert of any sorts. Once you have a solid content draft, then it comes to the visual part. And this is where most people struggle, because the most common concern I hear is that people say, I can't draw. I'm not creative. I'm not an artist. I can't draw. It won't look nice. Now, what I'm about to say is a bit of a hippie thing to say, but in the end, we're all artists. We are all musicians or dancers or painters. The one thing that differentiates some people from others is that they just go out there and try, and they are not afraid of, negative feedback. Mostly when people say I can't draw, it's not true. Obviously, everyone can draw. What people mean is, I'm afraid that what I draw isn't good enough that people appreciate it or something like this. So that's a very important barrier to get out of your mind. It's not that you cannot do it, but you worry that you won't get it right and that people won't like it. So this is the hurdle people often encounter. I think if you want to do it, then either try to get a little bit of, let's say you want to make a nice abstract with Illustrator. Then sit down for one day, check out Adobe Illustrator tutorials on YouTube, and you will be equipped good enough to make a reasonable graphical abstract. If you really are not into illustrations yourself, then I recommend to go to online tools. BioRender would be the most famous ones, which is paid, although, so you also have to pay a publishing fee for taking their images for graphical abstract, let's say. There are other platforms which are basically more open source libraries of scientific icons. This is a good starting point. And having some base skill in Illustrator that you can take these existing icons, which are free to use, and modify them a little bit to your needs, should get you to a solid point. But so to summarize, coming up with the content, everyone can do that. And the best thing to be good here is testing it with your peers. The other thing, if you believe in yourself a little and you really want to, you will manage to put together a decent looking graphical abstract if you are willing to learn base set of skills. Yeah.
I want to ask you about color. This is something I do not understand. I know when I see beautiful combinations of colors, how they work together.
Yeah.
But when it comes to me choosing them, it feels like there's infinite number of colors, right? And again, to put them together so that they work in a particular, let's say, branding way. When you see in Nature or Science or Cell paper, these are all so beautiful, guided by these wonderful illustrators. They all look so consistent, like just the membrane thickness and the little bit of a shadow that they add on these particular points. It makes you feel like this is all part of one book. Can you tell me some principles of design, choosing colors, choosing where to put, how to organize your proportions, things like that might be of practical use?
So for choosing colors, I recommend getting some inspiration from existing figures that appeal to you. If you don't want to start from scratch, there are basically two, I don't want to call them rules because it's more like guidelines. One is, in some fields, you have a particular preference for coloring certain elements a certain way. So this might be very field specific, and here it's helpful to, just look at existing reviews. Like you mentioned, the Nature review journalists. They have in-house illustrators, and they do a beautiful job in having this very consistent style of depicting figures. So get out there and get some inspiration from existing figures that you like. And you don't have to take the color scheme one-to-one, but that's often a helpful guide to set out what general colors you take, because you don't want to overdo it. You don't want to be all over the palette. You want to have one or two main colors and then one or two accent colors. That's usually enough. There are also tools to make nice color palettes. One is called Cooler, so Coolers. This I can recommend. This I use for most of my designs when I don't know which color pattern to go or color palette to go with. This is a nice online tool that just generates instant color palettes for you, and you can pick the ones you like. One important aspect that it's not obvious to people, but which makes a big difference when coloring spaces, what also all these nature review designers do, you have an object. Let's say you have a cell and a nucleus, very simple. You don't make the cytoplasm one plain color and the nucleus another plain color, but you use gradients. So you do have one main color, but let's say the cell will on the edges be a little bit darker than on the inside. So this is something you can do in Illustrator or Photoshop. It's not tricky. You have to get a little bit familiar with how this works, but use gradients for the fills. Already this will make such a big difference from, more like plump or non-elegant singular colored areas to something which looks a little bit more three-dimensional would be too much, but it gives it a little bit of depth and it makes it look more interesting. So stay in the lighter range and use gradients for most of the films. That would already be, makes a nice touch. Then other things which come to my mind is the outline. So whenever you have an object, you usually have a stroke or an outline surrounding it. The go-to choice is black. And black is stark contrast. What usually looks much more elegant if you go with some kind of gray, which still provides enough contrast that you really have an outline for your object, but it's not in your face. So use thin and more gray anthracite colored outlines rather than black. One thing I often see when people are not so trained in visuals is because you asked about layout. Things tend to be a little bit too cluttered because people put a lot of stuff and they put it all over the space so that you have something happening at every area of the image. There's this concept of negative space or white space. Not having something in certain areas will direct your attention to the areas where you want the attention to be, where stuff's happening. So avoid cluttering. Having empty space here and there is important because it gives you a balance on what to focus on, where's the action happening. Then for the general choice of colors, pastels. I like pastels because they are more subtle. It goes again in line with not having black outlines or black text. I find this also goes for presentations. Black text is high contrast, very much in your face. Gray is more subtle and somehow more pleasant for the eye to look at. What I sometimes have to think of, the comparison, some people talk very loud and you understand them, but it's not pleasant to listen to them. And instead, if you talk with a lower voice and a lower volume, then people will still understand what you say, but it's just more pleasant to listen to. So instead of being loud with your colors, try to be a little bit more subtle. It will be more pleasant for the mind to look at it.
What about, so this is all great advice for principles of illustrating figures. Well, what about if I'm giving a presentation with a PowerPoint? Do you have any slide design tactics or tips?
Yeah. So my favorite tip is maintain optical balance. This is very important, but I feel it's not such a well-known concept. So when you have a slide, and let's say you have 2 or 3 elements of the slide, right? Imagine your slide would be a board cut out of wood, and it hangs on a single nail in the middle top. And then you put your different elements, which have different sizes, on the board. You should put them in a way that if it would be physical objects cut out of wood, that the board would still hang straight. Then you have optical balance. So if you have different objects will have different weight depending on their size, and distributing them in a way or adjusting their weight that they are roughly the same size, so that the overall slide design is balanced. And if it were a physical slide, it would not tip to one side. This I feel is very important because it's somewhat unsettling when you have uneven distribution of objects on slides. So this would be the first main thing. The second thing is people tend to put too much stuff. So then the majority, when I'd work on presentations with clients, especially for graph applications, and I think I never said, this slide needs more stuff. That's never the problem. It usually is the problem that you try to make 6 points here. Either are all 6 points necessary to make so that your reader can follow the story or your audience can follow the story? And if so, then maybe split it up on different slides or just get rid of. Usually you can get rid of half of the points and still accessible to everyone who listens to you. Because I feel scientists are paranoid to be misunderstood. So the mistake they often make is to put every part of every information so that no one can accuse them of being too superficial. But this is actually not helpful when you want to tell a captivating story. So less is more would be the second main advice. And the third one is the text. And there I feel that it's a bit of a generational question. So the younger people get, the less text they tend to put on the slides. And this might be a bit of an edgy statement. In the end, I feel your style of presenting has to suit you. You have to feel comfortable. If you can follow tips. If you feel uncomfortable implementing them, you won't give a great presentation. Either way. But I feel that text is a crutch for bad presenting. So people often say, hey, yeah, but I need to put the text because if people don't pay attention to me, then they get lost. And then when they can read the text, they will know again what's going on. What too much text will inevitably create is an attention conflict between you, the presenter, and what's happening on your slide, especially if the slides are not animated. So I think one of the bad things you can do is you put a slide which has a lot of text and then you start talking about it. And then everyone will start reading the slide. They will not focus on you, but they will most of the brains will start reading the slides. So you create this conflict between people focusing on you and people focusing on the slide. And text it should be an aid to get your point across. It should not be the point you get across. You want people to focus on you. The much better presentation is where you are the center of attention. And your slide deck is basically your assistant in getting the point across. It's not you walking people through the slide deck, but it's you want the people to listen to you, and the slide deck is there to help you getting the point across, not the other way around.
This is something as an audience member that I struggle with, which is when somebody has text, meaning sometimes full sentences on a slide, and they're saying something else, especially if they introduce everything at once. So all of a sudden you get splashed with this slide with 3 sentences, 4 sentences on it. My immediate reaction is, okay, let me read that. But then the person out there is saying something. Maybe they're talking about those sentences. Otherwise, they would be reading them. But if they're saying anything a little bit different than what's written there, I am tuning out the person and focusing on reading. But then the person talking is bothering my reading. So I end up, I don't know, is it just me to this extreme extent, but I end up with that slide not understanding anything, even if the slide is fairly simple. So I definitely, as an audience member, I can understand this conflict of attention, which I think maybe is the biggest thing to solve. That you, during your presentation, you know exactly how the audience is perceiving. When are they looking at you because you're talking something? And when exactly are they looking at that graph or at this graph or at that little bullet point, whatever you made. So I think one thing that I really love about presentations that are well done is when they introduce steps in the slide, let's say. So if it's one graph, you introduce it now, and now I know exactly what to look at, right? Now I'm looking at it and you're talking to me about it. I can follow both of them. Image is visual. You are describing it. So the text is coming from your voice and the image is right there, allowing me to process everything. So I think that's a really good tip. Thank you for that.
If one thing, one thing I might want to add along the lines of what you just said, the power of animating things, and I mean with animation, I make them appear as you speak. This is extremely satisfying for the audience if if what you say is in sync with what appears on the slide. And for me personally, I never pull up a full slide. It's a title, and then I will say something, and then the element I'm talking about appears while I speak about it. And then when I'm finished making this point, then I will have the next element appear while I talk about it. And the synchronicity of what you say and what appears on the slide is very pleasant if executed well for the audience being walked through the process. And then I have one more thing I just thought of in the context of what makes a good presentation. Was your next question presentation related, or were we about to move on?
I was about to move on, so please add whatever you are.
Then, because this one is something I came across not too long ago, and it blew my mind in terms of how I think about what makes a good presenter or a good presentation. There's this concept of the hero's journey, the archetypical story or narrative structure that manifests itself in almost all stories we know. Are you familiar with the hero's journey?
I've heard of the concept, but please tell me more about it.
It's an amazing concept, and I'm amazed that it's not more well known. So when you like Harry Potter or Lord of the Rings or Star Wars or all these epic myths, they all follow the same narrative structure. You have an ordinary world, you have an underdog type of hero, they meet a mentor. So the Dumbledore or the Obi-Wan Kenobi or Gandalf, and the mentor helps the hero, the protagonist, to go through the journey they're about to make. So the hero goes from the ordinary world to the dark side, where it will encounter different challenges. It will have this final test, which is really the most difficult thing they would ever do in their life. And then after they pass the final test, they return to the ordinary world and they transform. They transformed internally and they transformed the external world. This is a very rough breakdown of the hero's journey. Now, what most presenters do is the cardinal mistake, which is they position themselves as the hero when they give a presentation. So especially in science, it's like, look at all the amazing shit we've done. Look at all the technically very challenging experiments we did, and here's this amazing conclusion we came up with. It has the emotional tone of, we are the hero, so listen to me, the hero, telling you how it's done or how it goes. In all of our personal individual stories, we are our own hero. Every human is their own hero in their own story. So people don't really want to listen to another person saying, look at me, look at how amazing I am. People want that you take them by their hand and that you guide them through an intellectual journey in the case of a scientific presentation. So your job as the presenter is, you are the guide or the mentor, you are the Gandalf or the Dumbledore or the Obi-Wan Kenobi of the situation. Your job is, if you do it very well, to open up a gap of knowledge in your audience, and then take them on the journey from the ordinary world, what they know, to the darkness, what they don't know, to the climactic moment where they learn something about biology which they didn't know before, and then they go back to the ordinary world and ideally learn something about biology. This is what good talks achieve, right? And your role is the mentor. You guide the audience. Your role is not hey, this is amazing what we did. Your role is let me take you on this exciting journey about biology. And if you understand this, and if you take this emotional position, then you did an amazing job as a presenter. Yeah.
I've never heard the hero journey as a tip formalized that way. So how would that look like practically? You said practically the mistake that people make is, I am the hero, meaning look at the science I did, look at this graph that I did, look at this result. I struggled with this and then I solved it. How would that in the little bullet points be transformed to put the audience in the hero journey?
Yeah. So for example, one concrete example that comes to my mind, you talk about a series of experiments that led you down like an empirical journey. And the difference between self-centered narrative and audience-centered narrative could be, and then we did this experiment and had this result, and so I wondered, could this be, so I did this next experiment. Instead, you rephrase it, and then we did this experiment, and this was the outcome. So now you might wonder, what does this mean for the other thing we are thinking about? And just the difference between, and then I thought, and then I did, to here I just showed you something, and now I assume that you were wondering about this other thing. So instead of having an I language and an I focus, focusing on the process that goes on in your audience's mind while they listen to your story. So yeah, shifting your general focus, you should always ask yourself, what will this elicit in the audience's mind, and which need will this bring up in the mind of my audience? And then good presentations are also often a series of, you open up a small gap of knowledge in your audience's mind, and then you fill them again. This is a very satisfying process to undergo. There's a gap not open in your mind, and it's filled again. Good talks do this with every slide, basically, but an audience-centric point of view. Always keep in mind, what effect does this have on the audience, and what is the audience thinking and feeling now, and what do they expect next, basically. Try to see it from the eyes of your audience. Yeah.
Before I move on to the last part of the talk, we have some more minutes, but I want to maybe ask just one last thing on this topic, which is, do you have any presentations that are already online that we could take a look at as an example of what's a good talk that you might recommend for practice? Yours or anyone else's might be a good example, like a TED Talk or something. All TED Talks are pretty good.
They're pretty good. I don't have one concrete example. If you are into this type of stuff, if you want to become a real good presenter, I have a hero who is my idol or my role model for getting these things right. It's Nancy Duarte. So Nancy Duarte runs an agency in the US, and they develop presentations. Nancy has a couple of books which are amazing. Also, they mention, or they touch a lot on these concepts, like the hero's journey in the context of presentations. So if you're interested in that stuff, Nancy Duarte is an excellent source to start educating yourself about the psychology, the psychological dynamics that unfold while giving presentations.
Amazing. Thank you.
A short notice from my side, when I started out, becoming a professional science artist, I did not expect presentations to be, I didn't consider presentations to be such a relevant niche topic. I like presenting myself. I also, during my active scientist days, I didn't mind giving a talk. And turns out that presentations are, there's a hot need for developing good presentations, because often presentations are high stakes situations, right? It's a grant interview. Do you get $1 million or not? It's a job interview. It positions you in your field as an expert or not when you go to that big Drosophila conference. So presentations there's a high demand, and I recently decided to develop professionally more in the direction of focusing specifically on designing solid presentations for situations like grant interviews or job applications, because there you can really make a difference by investing 30, 40 hours on designing a solid narrative structure and making a beautiful slide deck. The value you generate here, it's so worth it. So yeah, that as a personal note of, I also recently started to realize the significance of presentations also as a service if you are active in my my field, so to say.
Definitely. I think I have already seen in my early career how important a good presentation can be. It can be a difference between getting and not getting a PhD fellowship, or, some kind of award. And later in academia, getting a good postdoc, landing a job, wherever you go for an interview, you have to give talk presenting your research.
Yeah.
And very often some opportunities that you did not even know existed, like presenting at a conference where somebody who might give you some kind of opportunity will listen to you and they might approach you for a job, for a partnership, for whatever. So it's definitely a super relevant topic. Thank you for sharing that. If there are some folks out there doing science and thinking into side tracking into science art, what are some opportunities to explore? What are some resources on how to get involved, how to become a scientific illustrator?
So many people in the field had a strong autodidactic element, I feel. There are some courses available, but to my knowledge, there's no single resource I would recommend, which, gives you holistically all the different aspects you need to learn. I think part one is the technical aspect of knowing how to use your tools. This can be Photoshop or Illustrator. I'm a big fan of Photoshop. I love it. Here, this is how I personally learned how to use the tools. I listened to a couple of YouTube tutorials, but I never was good at learning something for the sake of learning it. I needed a project where I wanted to accomplish something, and then if I just wanted it badly enough, I would find a way to get there. So one tip would be, if there is a project, let's say your lab publishes a paper and they can submit a cover proposal, or a colleague asks you to, illustrate a header or homepage, a header image for the homepage, just commit to the project and you will figure out how to get there if you want it badly enough. YouTube tutorials for basics of Adobe Illustrator or Photoshop make a great help. But be technically fearless, and even if it's going to be frustrating at the beginning for learning how to use the tools, just wrap your head around something that you want to do and try to do it. And it will not be perfect the first time. It will also not be perfect the 10th time. For me, I don't know, I did a couple of hundreds individual illustrations. I'm still not, there's still always room for improvement when I see it. But just try it. And if you want it badly enough, you will find a way to get there. That would be number one. Number two is, if you do a couple of things that people like, then automatically people will come up with requests. So for me, it was in the beginning, I designed really for fun. I was drawing some stuff and put it on the lab fridge, something like this. And then somebody sees it and like, oh, cool, you did that. Hey, we have a paper coming out and need a cover. Why don't you give it a shot? So put out some things, and it doesn't have to be a lot, but people, if they like it, they will follow up and ask you for, if you want to work on, illustrating their stuff. In the beginning, you probably do it for free. For me, it also took a while to realize, hey, this is actually a valuable service. But in the beginning, don't worry about, doing these things for free to get some experience. The third important point where some people struggle, and I also had to grow into it, is get out there and share your work. Twitter and LinkedIn would be the two platforms that come to my mind. Twitter took an interesting detour the last 1-2 years. I'm not the biggest fan anymore. For me, LinkedIn would be the main outlet to really reach out to my target audience. But get out there and share your work. In the beginning, people often have a certain resistance to sharing stuff on social media because it feels pretentious, or they don't feel comfortable putting themselves in the spotlight. In the end, you're just sharing something that's important to you, and maybe it's of value to someone else. So when you want to get into scientific illustration, do some stuff, make sure to share it, and then opportunities will arise. Then I think you can find your way. By sharing, you can also develop a feeling for what resonates with people, what do people appreciate. I'm not saying that you should only draw what people appreciate, but it serves as a good guide when starting out for what type of my work is appreciated, which will also translate to, is there a market for if you really want to make a living with this. But yeah, get out there, share your work, and stay open-minded for opportunities that arise. And even if you have an opportunity but you don't know how to do it, then if you want it badly enough, you will figure out how to do it on the way there. Yeah.
Definitely LinkedIn is a great place. This is where I have seen your work and got blown away by it, by your beautiful illustrations for abstracts, for journal covers. Like those are some of the most beautiful covers I've seen, honestly, which is why I contacted you to have this chat, because you're obviously demonstrating that you're an expert in the field, even if you don't like the word expert. But it's just beautiful. I was fascinated and I wanted to have this conversation. I'm trying to find a good closing question for these episodes, and I can't think of anything better for now other than the cliche, but still useful. What's the advice that most impacted you so far in your scientific or illustration career?
Good question. Good question. I have to think a little bit about it. So one came from a PhD supervisor, Luisa Coachella. She had a lab here in Vienna. Now she moved to Johns Hopkins. The elegans geneticist, beautiful work, and a really, really cool person. So I'm grateful that I was able to do my master and PhD with her. And at the time where I felt a little bit lost during my PhD, I asked Luisa about I was sharing my doubts regarding academia and potentially also going on to scientific illustrations and what I feel I should be doing and what my natural self is drawn towards too. And Luisa, who is a hardcore academic, really hardworking scientist, and I admire her for her stringency, was very open-minded, always very supportive of my art, and it wasn't one statement that she said, but it was an attitude that she transmitted, which she would have wished for me to continue academia, I think, like most PIs do. But in the end, stay true to yourself, and if it makes you happy, you're probably gonna do it well. And there are many different ways of contributing to the overall scientific enterprise other than classic academic research. So if you feel that your heart is with the art and that's an exciting field, go for it and you can make a difference. And what I feel now, that because I still love science in the lab, I'm not bitter about academia. I think it's amazing what people do there. And I see that my way of contributing what science does is to craft a story to get out the information we keep generating. And that feels beautiful, but it's important to me also as why a general motivator. If you stay true to yourself and follow up on things that really excite you, you find a way how you contribute in your very own individual way to the overall enterprise of human culture. So don't worry too much about straying from the canonical path. If you're really into whatever alternative path you choose, then chances are that you will contribute something meaningful, and maybe more meaningful than if you would have ventured the classic path.
Philipp Dexheimer, everyone. Philipp, it's been a pleasure talking to you. Where can people follow your work, book a call, request a service, find you overall, and get in touch with you?
So LinkedIn would be the platform I'm most active. My homepage, philippdexheimer.com. It's also the when you want to get an overview of my work, it's the place to go. And there's a small contact form on my email address, so just drop me an email. I'm invariably happy to discuss new exciting projects and would love to get in touch with you if that might be something of interest for you. And hey, thank you, Kenan, for this very cool talk. It's always nice because I feel it's a shared exploration of some topics. So I also learned something today and appreciate it very much that you took the time. It was a pleasure to talk to you.
Pleasure talking to you too, Philip. Until next time. Bye, everybody.